The first step of urea cycle is the formation of carbamyl phosphate, and this step occurs بالميتوكوندريا of the liver. First, السبستريت هو الأمونيا plus CO2. الإنزيم هو carbamyl phosphate synthetase 1. البيوتين هو الكوإنزيم for this enzyme. Consume 2 ATP and lead to formation of carbamyl phosphate. The activator of this step or the activator for this enzyme هو N-acetyl glutamate. Positive allosteric, it means bind with a site other than the active site of the carbamyl phosphate synthetase 1 enzyme. So, الأمونيا بينتجينها من الجلوتاميك acid، يعني it means من الأوكسيداتيف deamination of glutamic acid. CO2 من الكريب سايكل، عندنا 3 خطوات بالكريب سايكل produce CO2. الفوسفات والانرجي from ATP. The second mitochondrial step of urea cycle is the formation of citrulline by reaction of carbamyl phosphate with ornithine. This reaction catalyzed by ornithine transcarbamylase and form citrulline plus inorganic phosphate. So ornithine and citrulline are both non-standard amino acids. That means not present in any type of proteins, only formed by urea cycle. The third step of urea cycle, formation of arginosuccinate. This step in cytoplasm of the liver. Use 1 ATP. Citrulline with aspartic acid lead to formation of arginosuccinate or arginosuccinic acid. Same. This reaction catalyzed by arginosuccinate synthetase enzyme. It means will consume 1 ATP. فالخطوة الأولى صارت 2 وهذا الخطوة هي اللي إذا ننتبه على الخطوات that consume ATP. Number 4, cleavage of arginosuccinates. This step occurs in cytoplasm of the liver. Arginosuccinic acid will cleaved by arginosuccinase or arginosuccinate lyase, the same, and converted to fumarate and arginine. What is the fate of fumarates? The final step of urea cycle is the cleavage of arginine. This step also occurs in the cytoplasm of the liver. Arginine by arginase enzyme and addition of water molecule lead to formation of urea plus ornithine. The overall reaction of urea cycle represented by this simple equation. Aspartic acid with ammonia plus CO2 plus 3 ATP lead to formation of urea plus fumarate plus 2 ATP and AMP 2 inorganic phosphate 1 pyrophosphate plus 3 water. This slide shows the simple explanation of urea cycle as a diagram. Liver هو the main site of this cycle. We have 2 mitochondrial steps, which is the first one ammonia and CO2, and you should know the source of ammonia and CO2. Converted to carbamyl phosphate by carbamyl phosphate synthetase 1, consume 2 ATP. The second step in mitochondria, reaction of carbamyl phosphate with ornithine by ornithine transcarbamylase lead to formation of citrulline. Then the first step in cytoplasm, citrulline with aspartic acid lead to formation of arginosuccinate by arginosuccinate synthetase enzyme. It means consume the third ATP molecule in this cycle. Then the arginosuccinate by arginosuccinate lyase cleave to arginine and fumarate. Then arginine by arginase will cleave to urea and ornithine. Urea excreted in the urine while ornithine return back إلى الcycle. طبعا خطوات الميتوكوندريا ننتبه عليها وخطوات السيتوبلازم source of ammonia ال CO2 number of ATP consumed المصدر الأسبارتيت and so on. طبعا the significance of this cycle is to get rid of ammonia because it is toxic, especially for the brain. The activity of urea cycle is regulated at two levels. Dietary intake is primarily proteins that lead to much urea production and prolonged starvation lead to breakdown of muscle proteins and lead to increase the production of urea by this cycle. It means الفاكتورز اللي يسوي اكتيفيشن لهذه السايكل. اما اذا كان زياده او برايمرلي نعتمد على بروتين فاكيد اليوريا سايكل راح يصير لها stimulation or activation. prolonged starvation راح نعتمد على البروتين اللي موجوده within the muscle and breakdown of this protein lead to increase the production of amino acid, then the catabolism, and then activation of urea cycle. So the summary of urea cycle, the urea cycle consists of 5 reactions, 2 mitochondrial and 3 cytosolic. The cycle converts 2 amino group, 1 from the ammonia and 1 from aspartic acid, and a carbon atom from CO2 to relatively non-toxic excretion product اللي هو urea. This reaction requires 3 ATP and lead to formation of 2 ADP and 1 ANP. Now this slide shows the urea cycle in the form of table form. Represent the steps, reactant, products, enzyme, and location. This is another method to explain urea cycle. This is up to you. As you like, any method is right. Now, the relationship between urea cycle and citric acid cycle. First, CO2 need for urea cycle from Krebs cycle. ATP also from Krebs cycle. Ammonia from glutamic acid by glutamic dehydrogenase. Aspartic acid from oxaloacetic acid by transamination with glutamic acid by aspartate transaminase enzyme. And finally, fumarate will convert it to oxaloacetic acid in Krebs cycle.